Hey everybody, what's up? How's it going? And welcome to the second respiratory lab. So, we're going to talk about some respiratory physiology things today. And the first thing we got going on here is this bell jar apparatus. So, basically, what this thing is, it's kind of like a representation of the respiratory system. So notice we got these balloons here. Each of these balloons would represent a lung, and the stopper up here would be the trachea. The plastic bell surrounding the lungs, which we see right there, would be the thoracic wall. And there's a rubber sheet at the bottom, which is kind of hidden, but you see it kind of like wrapped around, would be the diaphragm. And this bell jar model lets us see how pressure and volume changes can occur and see how air can move in and out. So let's talk about it a little bit. Here we see another view of it, just making it easier to see that diaphragm right there. The rubber sheet represents the diaphragm. And there's a little handle that you can push up or pull down. And basically what's going to happen is if you pull down on that rubber sheet, you're going to increase the volume, okay? And by the way, basically pulling down means the diaphragm is contracting. That's what you're representing here. If you're pulling down, the diaphragm is contracting. And what that's going to do is that's going to increase thoracic volume. Thoracic volume being the volume in the entire bell jar. And if you increase thoracic volume, you're also going to increase lung volume. And according to Boyle's law, if you increase lung volume, lung pressure is going to decrease. And what that means is lung pressure becomes lower than atmospheric pressure. And when we've got this pressure gradient, air is then going to flow into the balloons. In other words, the lungs. So that's what we're representing here, folks. By pulling down on the diaphragm here, we're representing diaphragmatic contraction, making the bell jar volume increase. That's what thoracic volume is. Making the lung volume increase. That's what the balloons are. Making the lung pressure decrease, and then air goes in. So we can model what's going on in our own body with this neat little apparatus. Now, similarly, if we push the diaphragm up, we push the rubber sheet up, that represents the diaphragm relaxing. So when the rubber sheet goes up, the diaphragm is relaxing. The bell jar volume, in other words, the thoracic volume, is decreasing. That means lung volume is also going to decrease, which means, according to Boyle's law, lung pressure is increasing, and that means it's becoming greater than atmospheric pressure. And when lung pressure is greater than atmospheric pressure, when we have that gradient, air flows out of the lungs, the balloons, in other words. So make sure you understand and know these two sequences. So I might ask you to put things in order on the test. Okay? All right. So we're going to play with these things in lab. Gently, of course. They're kind of neat. We can see how pressure and volumes change and how they relate. Now, got a couple questions for you. Pause the video, do all the labeling, answer these two questions here, then start it up again and keep on going. Now this machine is a spirometer. This is a fancy spirometer right here. It is a device that can be used to measure volumes of exhaled air. And there's actually a bunch of different volumes that we label. Okay, the volume of air that you're usually breathing in and out, the volume of air that you're usually bringing in and out is called your tidal volume. So the volume of air you are breathing in and out is the tidal volume. Sorry, I was getting kind of crazy with my squares there. So like what you're doing right now, normal in, normal out, that's tidal volume. Now, bear in mind, a typical value for your tidal volume is about 500 milliliters. Okay, obviously, you can breathe in more air. So let's say you breathe in normally, that would be tidal, but then you keep breathing in. That additional volume of air you're breathing in, above and beyond a tidal inspiration, that is called your inspiratory reserve volume. So it's like you breathe in normally, quietly, and then you take in more air and you fill your lungs. Okay, similarly, you could also breathe out more air than you can normally. Like right now, you're breathing out normally, right? You could then and keep going. That extra volume of air above and beyond a normal tidal expiration is called your expiratory reserve volume. Finally, there's something called the vital capacity. Not finally, we got 3 more to go here. Next, there's something called the vital capacity. The vital capacity is the maximum volume of air that you can move in or out of your lungs. So let's say you emptied your lungs as much as you could. You can never truly empty them, okay? But let's say you breathed out as hard as you could. If you breathed out as hard as you could, you would breathe out your tidal, your normal, plus your expiratory. Then, if you filled your lungs, you would bring that expiratory back in, bring the tidal back in, and then also bring the inspiratory reserve. So the trio here is equal to your vital capacity. So vital capacity is the maximum volume you can move in or out. And we could set up an equation. Yes, we can do some more math here. Vital capacity is your tidal volume plus your inspiratory reserve volume plus your expiratory reserve volume. So if we combine these three things, that's the max amount of air we can either move in or out, and that is called the vital capacity. And vital capacity is going to depend on a bunch of things. I think I'll talk more about that in the next slide. All right, another thing we got to be aware of is something called residual volume. And I just hinted at that a moment ago. Residual volume is the amount of air still in your lungs after you do a max expiration. So remember I said you can never truly empty your lungs. There's always leftover air, and the leftover air is called your residual volume. Now finally, here we get to finally. Finally, we have the total lung capacity. This is the maximum amount, the max volume of air your lungs can hold. So if you fill your lungs with air, totally fill them, you'll have achieved your total lung capacity. And as you might expect, total lung capacity is actually that residual volume, the amount that you can't get rid of, plus the maximum amount you can bring in, which is your vital capacity. So we got to know these two equations here. We have to commit those to memory. Okay? All right. In this beautiful graph here, Notice I got the normal amount going in and out. That's my tidal volume right here. In and out. And that's usually about 500 mils. Then we got the extra air somebody's breathing in. That's the inspiratory reserve. Then they breathe that inspiratory reserve out. Then they breathe out their tidal volume. Then they keep breathing out, and that's their expiratory reserve that they're breathing out. The amount left over is the residual volume. Now, notice that these three, inspiratory, tidal, and expiratory, that's what you can move into / out of your lungs. And that trio is your vital capacity. And in vital capacity plus the residual volume, the amount you can't get rid of, that's your total lung capacity. There's a couple things on here that we're not going to do, so don't worry about those guys. Okay? All right. I will make you do some basic math equations. But it's like going to be 2-minute math, okay? So don't sweat that. Also, I want you to understand something. Vital capacity is going to be predictable based on your age and height. So if your height increases, so increased height, let's get rid of that, all caps. I can't spell. Increased height. Increased height is going to mean most likely I've got longer lungs, and that means I'm going to have an increased vital capacity. What about if I get older? As I get older, what's going to happen to my vital capacity? Well, as I get older, my thoracic cage, my lung tissue, everything gets less stretchy, so it's harder to expand and fill those lungs. So with increased age, I'm going to get a decreased vital capacity. And we'll actually play with some ways to predict this in lab. Similarly, I can predict residual volume. And again, if I'm taller, increased height, I'm going to have longer lungs, which means I got more room for leftover air, which means I'll have an increased residual volume. If I'm getting older, all right, I lost my train of thought there, got interrupted, which is totally cool. It happens. What was I saying? Increased age. As I'm getting older, increased age. Again, I'm getting less and less thoracic and lung flexibility, so it's harder. My lungs are less elastic. It's harder for me to get rid of all my air, which means I got more left over. So kind of surprisingly, increased age, increased residual volume. So make sure you know these relationships too. Make sure you know these equations, the definitions, and these relationships. Okay? All right. We're almost done. This is going to be a short, nice and short video. The last thing we can talk about is another job of the respiratory system. This is like its unsung job. The respiratory system controls pH. It says here, recall from lecture. Maybe we haven't gotten to that in lecture quite yet. But carbon dioxide is going to undergo this reaction in your blood. In your blood, carbon dioxide combines with water, and that creates HCO3 minus. HCO3 minus, I know you remember from chemistry class, right? HCO3 minus is this chemical compound called bicarbonate. And not only do we get bicarbonate, we also get a hydrogen ion. So what we notice is that CO2 levels in the blood, because of this reaction, CO2 levels in the blood affect hydrogen levels and thereby affect pH. Remember that a nice way of having an equation for pH is pH equals the log of 1 over the hydrogen concentration. With what we got to notice here is that as the hydrogen concentration increases, the pH decreases. So of course, if our pH is 7, that's going to be neutral. Less than 7 is acidic, and greater than 7 is basic. Normally, our pH is between 7.35 and 7.45 in our blood. So normally our pH is a little bit basic. Now, hopefully we'll have a chance to do this experiment in lab. Basically what we do, what we're going to do is we're going to exhale through a straw, like blow bubbles, into a phenolphthalein solution. And by the way, phenolphthalein is going to be the greatest word to spell on your exam. So we're going to exhale into a phenolphthalein solution. And phenolphthalein is what's called an indicator dye. And it is pink when it's in a clear solution. Sorry, it's pink when it's in an alkaline or basic solution. And then it becomes clear when the solution's pH changes and becomes acidic. So what we would do is we would take a straw. We would put a straw in here. Boom, boom, boom. There's a straw. It's a horrible looking straw, I know. And then we'd have a person blowing into that straw. I'm going to get rid of that straw. I don't even like that straw. Can I make a straw like out of a square? Not really. That's crappy too. What's this? It's a line. Can I draw? Nope. All right, there's, what am I doing? I gotta just keep going. I'm wasting your time and wasting my time. We're going to put a straw in here, and we're going to blow bubbles in this solution. When we blow bubbles, we are adding CO2. It's going to react with the water in here to form bicarbonate, and importantly, to form hydrogen ions. So the more bubbles we make, the more hydrogen ions are going to be produced, and the more the pH is going to drop. And eventually, it'll just all of a sudden go from being this nice pink color to being clear. All because we blew bubbles into it. Okay, so the whole point is CO2 levels affect pH levels. So if you are hypo or hyperventilating, breathing slower or faster, that can impact your blood pH. All right, that is it. We are done. 18 minutes for this video. And I'll see you next time for the digestive system. Maybe I'll see you in lab or open lab. Let me know if you have any questions, as always. Bye-bye.